Welcome to the In Vivo Imaging Success Knowledge Sharing Video Forum. I'm Alexandra Delille and Director of Technical Applications at PerkinElmer for In Vivo Imaging. Today we will discuss the advantages and methods of in vivo fluorescence and cerenkov imaging of antibodies, proteins, and peptides. If you have questions and want to explore further, feel free to email me at alexandra.delille@perkinelmer.com. Optical imaging is a valuable methodology for preclinical pharmaceutical development. It is low cost, high throughput, with high sensitivity and translational to human medicine. Let's start with some examples of peptides, proteins, and antibody imaging. Here you can see an example of antibody tumor targeting. A TD tomato expressing PC3M tumor, pseudocolored in orange, is targeted with a fluorescently labeled therapeutic antibody, pseudocolored in blue. HER2-Sense is a fluorescently labeled humanized antibody based on the commercial therapeutic antibody trastuzumab that specifically targets and binds to HER2-positive tumors, such as the SCOF3 ovarian cancer cell line. Best signal to noise is attained at 24 to 48 hours post-injection. In this case, MDA-MB231 cells were implanted by intracardiac injection into nude mice. Multiple metastases, including bone metastases, were formed within a few weeks post-implantation of cells. The image on the left shows the bioluminescent signal. Avastin conjugated to CF750 was injected intravenously, and the fluorescence image shown on the right was taken 24 hours post-injection. The antibody conjugated to the dye shows great targeting to the metastatic sites. The RedEject integrin 750 probe is a fluorescently labeled cyclic RGD peptide that binds alpha V beta 3 integrin, enabling non-invasive fluorescence imaging of, for example, U87MG luciferase tumors in nude mice. Here is an example of a pharmacokinetic cross-validation of a dual-labeled Cy55 and copper-64 not-in peptide probe in a murine human tumor xenograft model. Plots of tumor-to-background tissue ratios correlated well over time, as published by Kimura et al. In Bioconjugate Chemistry. In vivo optical imaging methodology, what does it take? First, we need to label with a fluorescent dye. Labeling of peptides, proteins, and antibodies with a fluorescent dye is straightforward. You can label them with an easy-to-use kit. Near-infrared fluorochromes, specifically optimized and validated for in vivo imaging, are available. You can choose from amine-reactive near-infrared fluorochromes for labeling via an NHS ester linkage, thiol-reactive NIR fluorochromes for coupling via malamide chemistry to label free cysteines or thiol groups. Non-reactive control dyes of the same wavelength are also available. Treatment groups and imaging views. In order to determine biodistribution, it is important to design a study with the correct treatment and control groups. Traditionally, we have a blank control group, a non-reactive dye-only group, a block target group, the fluorescently labeled antibody of interest group, and a non-specific IgG labeled antibody group. In 2D settings, both dorsal and ventral, and sometimes lateral images are taken. This data is from the group of Dr. Zhu et al., published in Molecular Pharmaceutics. In this study, the same mice are imaged longitudinally at various time points in both dorsal and ventral positioning up to 96 hours. Regions of interest in the tumor and liver or kidneys are quantified and data is plotted over time. In vivo methodologies still need ex vivo validation. Tissue fluorescence can rapidly be quantified and validated in ex vivo tissues. Simply take your last in vivo image, necropsy the tissues and organs, and image the tissues ex vivo with the in vivo optical imaging system. A challenge faced in monoclonal antibody preclinical development is whether the antibody is specifically binding to the relevant target in the tumor tissue. Although we can observe accumulation of the fluorescently labeled antibody in the tumor with in vivo imaging, we still need ex vivo validation. With the nuanced spectral and mixing microscope camera, equipped with a liquid crystal tunable filter, we can not only see where the fluorophore is inside the tumor in ex vivo tissues, we can also co-localize the antibody binding to the antigen with up to 5 molecular markers simultaneously within a single tissue section. As described by Correa et al., serum stability is an important assessment in the therapeutic antibody development decision-making process. The lab chip GX2 offers a high-throughput analytical technique with very good precision and sensitivity. Fluorescently labeled antibodies can readily be analyzed from blood. There is no need for gels, and electropherograms give a reading in just seconds from a microfluidic chip. What is the sensitivity of fluorescence imaging? Fluorescence imaging sensitivity in deep tissues is greatly enhanced by methods of transillumination and offers picomole sensitivity. One can detect down to 0.4 picomole 800 nanometer quantum dots deep in the lungs of a mouse. Translational imaging. Optical imaging of radioactive probes by Cerenkov light. Prior to clinical trials, an antibody can be tagged with a clinical radionuclide. For translational purposes, the distribution of this radionuclide can be screened in lab animals using an IVIS in vivo optical imaging system, which has the sensitivity to detect the Cerenkov light emitted by beta-positive and beta-negative radio tracers. Cerenkov light is visible light produced by charged particles. This is what happens when radionuclides decay. A proton inside the nucleus turns into a neutron and emits a positron and a neutrino. The positron travels 1 to 2 millimeters, slows down, and combines with an electron and produces 2 gamma rays. PET imaging detects the gamma rays, while optical imaging detects the ejected positron or electron if their relativistic traveling time is faster than the speed of light in the medium. Although this Cerenkov light emission is faint, it is in the orders of magnitude of bioluminescence, and since all our IVIS imaging systems are cooled to -90 degrees Celsius, the IVIS is capable of detecting and quantifying this Cerenkov light emission. Both beta plus and beta minus particles generate light, and therefore various PET and SPECT radionuclides can be detected by optical imaging. Here you can see an example of F18-deoxyglucose, sodium iodine 131, and an yttrium-90 RGD bombescent peptide, as published in an excellent review by Drs. Liu et al. In PLOS ONE. In a study conducted by Drs. Ruggiero et al., medical isotopes were imaged by PET and Cerenkov light imaging at equal doses, and both the qualitative and quantitative data were found to strongly correlate with immunoPET and biodistribution studies. Biodistribution of fluorescent antibodies, proteins, and peptides can be visualized in 3D tomographic reconstruction. By means of transillumination imaging, 3D ROIs, and tomographic reconstruction algorithms, we can quantitatively assess whole body biodistribution of fluorophores in 3D. The quantitative aspect is confirmed by strong correlations between in vivo and ex vivo tissue fluorescence data. This work was published by Drs. Vasquez et al. In PLOS ONE. Here is another example of a 3D reconstructive representation of Sci-Fi-5 biodistribution to deliver in co-registration with micro-CT. As a final note, I would like to quote supportive references from the literature. Drs. Kimura et al. Dual modality near-infrared PET imaging agents are promising for clinical applications in both deep tissue imaging and image-guided surgical resection. Further stating that copper-64 and Sci-Fi-5 uptake plots correlate well, demonstrating pharmacokinetic cross-validation. Drs. Fukukawa et al. Found that results of in vivo biodistribution of a monoclonal antibody assessed by both radionuclide and fluorescence imaging modalities were very concordant. Dr. Sun et al. Simultaneously evaluated 2 antibodies, each labeled with a fluorophore of a different wavelength, in one single tumor mouse, allowing to assess potential interactions that can affect pharmacokinetic and potential side effects. For Dr. Dean, the high-throughput, low-cost, short anesthesia and lack of ionizing radiation is a great advantage. When looking at the advantages of Cerenkov light imaging, Dr. Robertson found a statistically significant correlation between PET and Cerenkov and the 10-fold increase in throughput capability of Cerenkov versus PET imaging to be a significant plus. Drs. Ruggiero et al. Also stressed the advantage of the rapid, low-cost, high-throughput screening methodology of Cerenkov light imaging. Thanks for viewing this knowledge sharing presentation. Stay tuned for more videos. If you have questions, feel free to contact me by email at alexandra.delille@perkinelmer.com.